Hello, everyone. My name is Ian Fernandez, and today I will be presenting my selected article that deals with tracking temporal shifts in the human microbiome under short-term spaceflight conditions. Now, I selected this article because I'm personally interested in learning more about how biology reacts to the conditions of microgravity. In other words, you can probably say that I'm a space nerd. But yeah, let me take you through the introduction, give you guys a little bit more background on the study. So just to present the bottom line up front, space flight is capable of causing significant alterations in the human microbiome. And this is very significant nowadays more than before because now we have a rise of commercial space flight. So now what we have is what we're seeing here in this Inspiration 4 SpaceX mission, which I'll talk about in a few minutes, that is completely made up of civilians. And civilians with a lot of money are now able to take trips to space through a variety of different avenues, whether it be a private company uh, or private entity. Um, they're planning longer space missions for the future as well, because now the next frontier of space exploration research lies a little bit beyond the realm of the rocky planets. Instead of going to Mars or Venus, now we're going to be venturing out into the moons of Jupiter and Saturn to look for life, for example. And so this is going to require more intimate knowledge of the microbiome so that astronauts that are in space for a long time won't have to worry about succumbing to any kind of microbiome related illnesses. But I will tackle these points later on. I did want to highlight the research question, um, which basically purports how does short-term spaceflight impact the human microbiome? More specifically, its composition, gene expression, and interactions with the host immune system. Um, the authors of the selected study were able to reveal that there was transient shifting and persistent shifting of the microbiome during the 3-day short-term spaceflight. And I'll explain what it means in a couple of moments. But basically, they were able to find that spaceflight affected the microbiome. And this provided some insight for future space missions, more specifically, missions where space microbiome experiments can happen. So just to provide a little bit of background, and one of the reasons why I think this article is very exciting is because it takes a look at this opportunistic mission that sort of happened back in 2021, I believe. It was called the Inspiration 4 mission. It was not a NASA mission, but instead a SpaceX mission. To the right, you will see the four individuals that made up the crew of the Inspiration or I-4 mission, which was the first all-civilian mission to orbit. And so one of the things I wanted to remind you all of is the harshness of the microgravity environment. Space, I don't think I need to say much about it and about its harshness, but it is considered an extreme environment. It's constantly exposed to high levels of solar radiation. Microgravity affects human physiology in certain ways that it's not accustomed to because, as we all know, we evolved on Earth, which is an environment where there is constant gravitational force acting down on our body parts. But that is besides the point. I did want to point out that there were also previous studies that added on to this background that basically tells us that spaceflight can alter the human microbiome, and not just that, but it has implications for astronaut health. For instance, Mortazavi and collaborators was able to demonstrate with their 2024 manuscript that microbial physiology, genetics, and community composition can indeed be altered in the presence of spaceflight. We saw that there was a link between host-gut microbiota interactions and astronaut health with Gonzalez and collaborators, a separate study that was also published in 2024. But most exciting of all, I think, is the NASA Twin study, which was a previous microbiome study that was conducted on the ISS, in which a set of twins, they were both NASA astronauts, but one of them stayed on Earth, one of them went to the ISS for a long period of time, and they were able to compare microbiome effects in both environments on the principle that there was microbiome similarity from them being identical twins. However, there was some limitations to this study, which did provide some limited precedence to the current study. But this is mainly because of the small sample size. There was only one microbiome of one person measured. In contrast, this study is going to be looking at the microbiome samples of four individuals under multiple different time points. All right, so if you see the image at the bottom, you will see Richard Branson, which is one of the billionaires that has invested a lot of money into making this creating the space startup Virgin Galactic, and here he is in space, one of his spacecraft. But he's not your traditional NASA astronaut, and I just included this image here to demonstrate that it very much is possible to see this more and more common as time progresses. Is more humans being able to go to space a lot easier, and if you have the money, being able to travel to space. But with being able to travel to space, you have these different health effects that can happen that are linked to the microbiome. And in this case, the reason why there's so much urgency in paying attention to this kind of research and continuing to fund this kind of research is that with the more long duration missions that are planned for NASA astronauts, or in general for civilian science, microbiomes are going to start to become more and more important to keep in mind. Mortazavi et al. Emphasized this as well in their 2024 manuscript, because as we know, with the whole personalized medicine argument, everybody's microbiome tends to be unique in certain ways. It depends on a multitude of factors, and to try and dial down which factors are affecting what interactions in the microbiome is why more work needs to be put into this kind of field. It's also been shown in the past with previous work, according to the current authors, that shifts in the microbiome in space have led to some negative health consequences, such as some GI discomfort, skin barrier disruption, as well as the suppression of immune cells and the immune system. I also wanted to point out the ISS effect. And this effect was spoken about in the paper as well. And this was first reported in the NASA Twin study. And basically, the reason why this was conceptualized is because they saw that there were microbes on the ISS that developed antibiotic resistance without necessarily being exposed to antibiotics. And this can pose a problem if we don't continue to study microbial adaptations in space. All right, a little bit about the article itself and the goals of the study. So Tierney and collaborators set out to essentially track the microbiome of three different tissue sites. So in this case, it'd be the skin, the oral microbiome, and the nasal microbiome. I'll get into more detail in a moment. But basically, they wanted to track how the abundance and expression levels of all of the taxon identified with all of the metagenomics and metatranscriptomics. They wanted to see what these microbes were doing. They wanted to see who was there. And combined with this approach, you were able to kind of generate a story with kind of like what was happening amongst themselves ecologically, what kind of interactions could have been happening. But I wanted to mention that this is a largely observational study. That still requires a lot of additional validation and replication in order to really dial down causality and more accurate correlations. But we did want to also look at the correlations between these shifts in the microbiome during short-term 3-day spaceflight and the immune cell gene expression or immune system activity. And now let's get into the methodology. So the authors of the selected study designed this to span across multiple different time points. That is why this is a longitudinal study, actually. Longitudinal refers to metrics taken at various time points in order to track changes over time. This study in total lasted a total of 6 months, even though the space flight itself lasted only 3 days. Sample collection begun 92 days before launch and spanned up until 82 days after the spacecraft returned from its 3-day mission. The participants slash subjects of this study from which the samples were collected were 4 healthy Inspiration 4 crew members. The steps of the study for the methodology are shown on the left, but I will be going over them in more detail in the slides that follow. On the figure on the right, you will see like an overview of how the experimental design is looking like. This includes the 10 body sites that I will go over in more detail in the next slide. This is how they went about collecting the samples at each of these different time points, what they labeled these time points. They also swabbed the capsule itself, which is called Dragon, but it's not that relevant for the current presentation since we will be mostly talking about the metatranscriptomics and metagenomics with the 4 astronauts, not so much with the environment and the environmental samples. Following collection and sequencing with their Illumina high throughput technology, they utilized a very specific data processing workflow to be able to parse out the bacterial and viral taxa, as well as create functional profiles of the genes that are being expressed. And then lastly, they performed a statistical analysis in which they were able to categorize and assign statistical significance while also maintaining validity under all of the different statistical confounds, but I will be going over those in a few slides. Alrighty, so this goes more into detail into the sample collection. So the figure on the right will show you the 10 different body sites at which swabs were taken. These were metagenomic slash metatranscriptomic swabs total of 750 swabs were collected as a part of this study among the four healthy astronauts. These samples, as you saw in the overview, were collected at different time points, but already we are seeing that there are more body sites being sampled in this study when compared to the NASA Twin study, which only sampled three body sites of the one astronaut. So already we're seeing a lot higher sample sizes, a lot larger sample sizes here, which is going to increase the significance of the data. Also, these body sites were selected because they are representing each of the three microbiome types shown here on the left. And lastly, they did collect blood samples from the astronauts, and they did this so that they could isolate the peripheral blood mononuclear cell cells from these samples. They're like serum samples, and then they did this so that they can do the immune cell profiling as their final experiment as part of the methods. I'll go over that in a bit. Alrighty, so longitudinal sampling here, this is just going to highlight how the samples were taken at different time points. What you see in the top figure here is with this 92D, 44D, that refers to the days before the launch of the capsule. And this goes to show that they were able to be more robust with their data collection because of these different time points. It makes it so that you can definitely create a much more thorough picture of what's going on with these microbiome expression profiles and how they're really changing as a result of this spaceflight. So that can be seen on the left with each of the labels for the time points that you can recognize. And I wanted to mention that these swabs were not immediately sequenced on this spacecraft, since it was only a 3-day space mission. They stored the swabs after collection at negative 80 degrees Celsius, and then they brought it back to Earth afterwards for sequencing. The thing that they used to sequence was the Illumina NovaSeq 6000 platform. This sequencing technology is of the highest throughput when compared to the other sequencing technologies currently. That just means that they can simultaneously and very quickly sequence a large amount of reads. And in this case, the Illumina NovaSeq 6000 was used for their metagenomic and metatranscriptomic analysis, which combined creates that multi-omics approach that I mentioned earlier. And this was so we can sequence both the DNA and RNA present in each of the from each of the swabs collected from each of the body sets of the astronauts. Although the Illumina NovaSeq can be disadvantageous when it comes to de novo construction, since it is short fragment lengths rather than longer reads. It's like shorter reads. So normally you would have to kind of combine the Illumina technology with like a PacBio to enhance accuracy of the genome building. But in this case, the authors saw it adequate enough to just utilize the Illumina and then just utilize multiple databases to try and eliminate the confounds. So this is the data processing pipeline that they utilized. After the samples were collected and sequenced, they were trying to extract meaningful information from the raw sequencing reads. As you can see in the extended data figure 1 on the right from the study, there are several key steps involved in this pipeline. So they performed this quality control on the sequencing reads to first remove any low quality or erroneous data. And then they used a combination of short read alignment and de novo assembly, or other words, assembly from scratch, to identify the microbes present in each sample. So all the taxon or the WHO, the metagenomics or the DNA. And then they did a short read alignment involved, or I apologize, the short read alignment that they performed was involved in building these genomes from scratch without relying on a reference genome. And furthermore, they performed taxonomic profiling with the MetaPHI-N and GTDB databases because they were trying to parse out those bacterial and viral taxa. And then they performed their functional profiling with the metagenome assembled genomes that were yielded from the taxonomic profiling to understand the functions being performed by the microbial communities. And this involved identifying the genes present in each sample and measuring their expression levels. Right, for the statistical approach, the primary technique that was used was the linear mixed effects modeling, or LME modeling, and this was used to identify the bacterial species, viral genera, and microbial genes that were present in the sequencing data. And this was used to conduct an MAS, which is a microbiome association study, in which associations for each taxonomic rank were able to be determined with their algorithmic approach. False discovery rate was taken into account in order to prevent redundancies of multiple different database entries of the same taxon, for example. So they had to do this FDR quality control step as well to control for that. And then based on the statistical significance of these assigned shifts, which we'll see in a bit. They were able to categorize these values, or in terms of the number of taxon, or in terms of the genes expressed, they were able to categorize these data points into each one of these groups. Basically, what they were trying to look at with the multi-omics longitudinal study is if these changes persisted past spaceflight or if they just remained within the confines of spaceflight. So transient increases or decreases are temporary, and persistent decreases or increases are more or less longer lasting. Now I'd like to go over the results. Alrighty. So the first set of results I wanted to look at here was a comparison between the abundance and expression uh, shifts that were seen by the astronaut microbiomes following spaceflight. And so let me just break these down for you real quick. So in the figures that I will be presenting in the next slides for the results, you'll see a lot of blue and red. Wherever you see red, it means metatranscriptomics, which is the RNA or the genes that are expressed. Whenever you see blue, that refers to the metagenomics, and this is the total DNA that was found, which is linked to the species that are present, or the who is there, the abundance of the particular uh, genera or species or cells, and so on and so forth. So a couple of findings that we take from these figures, excuse me, are the following. So we see that mainly the changes that are statistically significant in these astronaut microbiomes, they're mainly occurring transiently, or in other words, they're mainly occurring within the confines of the 3-day space flight. And when it occurs transiently, you'll see it revert back to how it was before the astronauts went to space. So when it's a transient change, it goes back to how it was after they returned back from space. And there is no like long-lasting effect there. And so it's important to point out that the majority of these statistically significant changes are transient. As you can see, the majority of the blue and the red uh, that has been plotted here in terms of the number of significant hits correspond to the transient changes. And then in this figure on the left, you will see that when you plot the relative abundance of each of the microbiome identifiers that we went over last time, the majority of these peaks are happening during the FD1 and FD2 time points, which is the time points that occurred while the spacecraft was in space. Furthermore, I'd like to point out something called the discordance, or in other words, that any changes that we see in the amount of DNA won't necessarily equate to changes in the amount of RNA being transcribed. Um, and this has some significant implications that we will go over later on in the slides. But I did want to introduce that here because we do see that this blue and red are not the same. Excuse me again. And then I wanted to lastly point out this one finding from the oral microbiome. When you compare all of the body sites that were sampled, remember the oral one represents the oral microbiome. And what we see here is the most amount of microbiome shifts in general, whether they be abundance or expression shifts. We see the majority of them are happening inside of the mouth of astronauts. So let's dive deeper into each one of these microbiomes. So firstly, we will dive into the oral microbiome, which yielded some pretty interesting results. As we can see here, there's a pretty significant decrease and a pretty significant increase in, excuse me, in this bacterium and this bacterium here. So right now we're only showing persistent increases and decreases because the figure was too large to fit into one slide. So here we are seeing that for Algindivalis, which is associated with periodontitis, believe it's an opportunistic pathogen, just like this one at the bottom. This one is experiencing a decrease in expression of its genes following spaceflight, and it persists past spaceflight. So that's pretty interesting. And then here we have Eloscardovia omicolens. This is another opportunistic oral pathogen that is commonly associated with tooth decay in infants. But it's strange because we're seeing that its abundance is persistently increased following spaceflight. So that means that it's not necessarily transcribing any genes, it's not expressing any genes, there's no gene activity, but there is an abundance of these microbes that we have seen change after the mission was over compared to beforehand. And I also wanted to pause for a moment and just point out that we have certain graphs here in the middle of these architecture profiles, and that just represents the statistical significance of the decrease and the increase. So I also wanted to point out something that was in the previous slide, but you can see it more or less on this continuation slide here, which is a continuation of the same figure from the article, is that you're basically seeing a lot more metatranscriptomic shifts than metagenomic shifts in general with the oral microbiome. So basically, we're seeing a lot more of these taxon that were sampled expressing a lot more genes after being exposed to 3 days of spaceflight. Or they could be decreasing a lot more significantly in their gene expression. But the point is that we are seeing a lot more of this metatranscriptomic movement, so to speak, versus the abundance itself. Um, and I also wanted to point out on this slide here that there was a lot more transient increases in the abundance compared to transient decreases in the abundance. So that's another curious thing. We see that the microbes in the mouth of these astronauts, they're increasing in abundance while in microgravity, and this can potentially hint at how they may be able to propagate in this environment in microgravity, or in general, just while under spaceflight conditions. All right, moving on. We are going to be now looking at the skin microbiome. And the skin microbiome, remember, encumbered those 8 body sites that excluded the nose and the mouth that were previously listed. You can see them actually listed at the bottom here. And the first figure on the left represents the viral shifts, or the shifts in the viral genera. Um, and they were also assigned that one of the 4 categories based on their significance of their gene expression. Um, and so basically what we're seeing here is that during spaceflight, there were noticeable hotspots of viral activity, both with eukaryotic viruses and bacterial viruses, or phages. And you see this hotspot happening in the gluteal crease more so than anywhere else. So this hints at the fact that these viral, or sorry, these microbiome interactions or these shifts are more than likely body site dependent, because we can definitely see this here. And we also noticed that there is more or less just transient changes with the viruses and not persistent ones. Just something that drastically changed while the 3-day space flight was happening. Um, and then when we look at the first half of the skin microbiome figure on the right, we see some interesting results here. So firstly, we see that these Acinetobacter species are persistently decreased in the T-zone and the behind the ear area. And this happened even after space flight. And we saw a decrease, not in their abundance, but in their gene expression. Similarly, but actually kind of different, is this significant change in Lecnospira. And this is actually quite a unique finding in this figure, because this is the only significant persistent increase that happened in abundance. So for some reason, Lecnospira increased in number after spaceflight and during spaceflight compared to before. And again, this is from the skin microbiome. So this is a skin microbiota. And then you can also see some transient decreases that were listed here, but there was no metagenomic or abundance shifts involved in that, purely just expression shifts. But in the next slide, you'll see that the skin microbiome actually experienced probably the largest effect of this whole study. And that's that the majority of the members of the skin microbiome transiently increase. So of all of the skin microbes sequenced, we see an increase in both expression and abundance for all of these members and these associated body sites only during the window of spaceflight. And then something interesting I wanted to point out here in this figure, or this continuation of the figure from the previous slide, is that um, we have members that are more or less common or universal to skin microbiomes, like Corynebacterium, Acinetobacter, like we saw in the previous slide. And these are expressing, or sorry, these are showing expression shifts during spaceflight. And when you compare that to the abundant shifts that we see in microbes that are not so common to the skin microbiome, such as Mesorphysobium, this species right here, this species is not really universal to everybody's microbiome. It'll always depend on like the geography, a lot of different factors, but because we see that there is an increase in the abundance of a not so common skin microbe on the microbiome of the astronauts, during spaceflight, this kind of hints at the interactions that these 4 astronauts in close proximity are having with each other, and what interactions their microbiome members may be having with each other as well. Especially since this is the skin microbiome, and astronauts touch surfaces and they leave their microbes behind and everything that they touch. So I would anticipate that in this microbiome, you would get the most amount of movement as well among the microbes. I just wanted to demonstrate that here as well with the Myxorhizobium. and how you just see the metagenomic changes. All right, and then lastly, I want to go over the correlation that the authors were able to make to human immunity. This is the immune cell, the single nuclei immune cell profiling, in which they basically connected the shifts that we just went over with the gene expression of the following immune cells that we see listed on the top on the figure to the right. And then what we see here that's really interesting is that the oral microbiome uh, metagenomic and metatranscriptomic shifts seem to be correlating with the immune cell expression. Because what we see here is increase in abundance of the microbial genes. We see increase in expression of the bacterial taxon and bacterial genes. This doesn't have much to do with the viruses in this case. More or less, it's just with this group and this group. But we are seeing temporal immune cell gene expression, and this goes to show that the effects of space flight on the human microbiome may be having a more significant effect on the immune system than we previously thought. And also, I wanted to point out that microbiomes are not universal. Microbiome signatures are also not universal. And with this said, microbiomes are unique to the host in one way or another that we're still trying to elucidate. But because of these differences in each individual microbiome, a lot of confounds can arise when drawing conclusions from these types of data and trying to link these types of expression profiles with a correlation analysis to try and create some kind of causality. All I'm trying to say is that more work definitely needs to be done with human microbiomes in space before we can begin to parse out the exact factors that are causing these changes. Alrighty, I just wanted to bring it home with the bottom line up front once again. That space flight, after learning about the article, we've learned that short-term spaceflight particularly is definitely able to cause detectable shifts in the human microbiome, and thus it is able to affect it and affect it significantly. We saw that with comparison to the other microbiome samples, the oral microbiome had changes that were more persistent on average and more significant, more abundant on average than the other ones. We also really doubled down on the significance behind that association behind immune cells and immune cell expression with these particular gene expression and taxon abundance shifts that we saw. Uh, Definitely with the oral microbiome, once again, wanted to kind of highlight that as our key finding because we did see the most amount of shifts there. We saw the most amount of persistent shifts there. We saw the most amount of metatranscriptomic persistent shifts happening within their samples. And I just feel like that's significant. That's significant because there are still opportunistic uh, oral microbes that can definitely cause disease, periodontitis, for example, like we discussed before. And these opportunistic pathogens can infect astronauts that get tooth decay in space. And if there's not appropriate therapeutics to treat uh, said negative microbiome shifts, for instance, then that could spell bad news for the astronaut if they're on a long-term space mission. So with that said, there are health implications of this work, as I reiterated before. Not only that, but spacecraft design can be optimized to better suit astronaut needs if more work is done in this field, and we can better understand the contributions to microbiome changes, which could be, diet, hygiene, and environment other than spaceflight itself. But it's definitely worthwhile to learn more about it. So some key takeaways. Bottom line, once again, spaceflight significantly alters the human microbiome. Um, Skin microbiome we saw had a lot of transient changes, a lot of transient increases. So we can interpret that as the skin microbiome picking up new bacteria as the astronauts touched different surfaces and moved around the capsule. Viral activity was also seen to be increased in the gluteal crease during space flight. Again, more or less a transient increase and a transient change. Um, so with that said, the majority of these microbiome changes were mostly short term. Very little relatively when you take into account the big picture were persistent changes or persistent microbiome shifts. And then some future directions, as I had mentioned before, for this research, will most likely be a study done in a long-term space mission with perhaps more time points again with a longitudinal study design so that we can track it over time more effectively with the same people. And this way we're able to find ways to better understand the astronauts' microbiome so we can keep astronauts healthy. Alrighty, we're approaching the end of the presentation. Below are 5 questions that I've posed to try and fuel a little bit of discussion with regard to this, since it is a very new and up-and-coming field of research. I'm very curious to know everyone's input. So first question, given that the microbiome shifts observed in the short-term spaceflight study were largely transient, what are the implications for long-duration spaceflight? Do you think these shifts will become more pronounced or persistent over long periods of time? Yeah, what impact might that have on astronaut health as well? What factors could explain the disconnect between the metagenomic and metatranscriptomic changes? Could this be because of niche microbes? Could this be because some microbes are purposely not expressing genes because that is their role in that particular microbiome environment? I would certainly love to know your thoughts on that. Also, some curiosities arose about phage activity and how that may be able to impact these bacteria that are in the astronaut microbiome, since phages infect bacteria. Um, I'd also like to know if anyone can elaborate on the potential of implementing tools such as Minion or Epitumi, like we learned in class, for conducting these kinds of studies on the spacecraft in real time with minimal weight load and with similar, if not better, sensitivity and throughput compared to Illumina. So I would like to know your thoughts on that as well. And then lastly, how can this research be used to inform future spacecraft design? If there's any ideas you guys have behind the interior or the materials or the layout, potentially increasing the space within spacecraft to limit interpersonal contact between astronauts. Kind of curious to know what you guys thought about how we can devise different designs in the future. Alrighty, and then that's all for me. Thank you so much for listening.